Here we are, ready to embark on a new journey into the invisible world, the microscopic world. There are so many amazing things to see, but you need to know how to use the equipment and the items that are part of being able to view this amazing world that you've maybe never seen before. So microscopes vary in the different apparatus that they have on them, different components, but some of them are always the same. And we're going to talk about how to use a microscope, how to transport the microscope, how to actually put a slide on the microscope, how to make a wet mount in the future. So these are just some really basic skills that we're going to talk about today that as scientists, especially in agricultural biology, you need to know how to do. So without further ado, let's get started. This information on how to handle your microscope is in your safety manual in your binder. Okay, so you have come into class, so you need to get a microscope and get ready to get set up. This part right here is called the arm. This part down here, this big bottom, is the base. You'll carry it by the arm and the base. Always, always, always 2 hands on the microscope. Okay? And then you set it on the table. And when you plug it in, you want to make sure that this cord is not dangling. It's up out of the way so that somebody will not catch it and drag your microscope onto the floor. Another key important part is that when you unplug the microscope, that you hold firmly to the end of the cord where the plug thickens out. Pulling on the cord will dislodge the plug, and then the microscope will not be safe for use. Okay, so now you've got your microscope to where you're going to be doing your work. What's important in examining your microscope? So you're going to take a look at your microscope. You want to make sure that you don't tip it upside down, tip it around, because some microscopes have eyepieces that just fall right out. Okay, so you want to take a look at it. Does it look clean? Take a look at, is it on low power? Low power on these microscopes are going to be the red ring. The red ring is the lowest magnification, and that's where you have to start your focus. The red ring is the color of the ring on the actual lens. Make sure that it's on low power. The other thing that you want to take a look at is, this is where I have found terrible problems in the past. If you look at this space right here, this space, it's not equal. And when that space right there, the metal parts going out to our fine adjustment, when those are not equal, when you go to focus in on high power, it's going to be extremely difficult. So if you will get those leveled out so that they are about the same on both sides. So I'm going to turn this around again. You can now see that they are mostly equal. And that's going to help you focus. That's the key. That is the key for making your microscope be focusable beyond low power and be able to get very clear. So let's talk about all the parts. We talked about the base. Moving up from the base, you usually have the light source. Okay, and once your microscope is turned on, a lot of these have adjustment for the light source. You can see that that's going up and down in light. Okay, then we have, working our way up, we have right here, we have our fine adjustment. Moving our way up from there is this knob right here. On this one is a special one. So not always will they be set up like this. So when I turn this lower knob, you can see that it's moving this piece right here back and forth. That controls where my slide is underneath the lens. Then when I go to the larger ring, it will move my stage, that's this piece right here, it will move my stage forward and back. So this one on the bottom moves my lens left to right, and the larger one closer to the stage moves my stage forward and back. And that's so that you can find what you're looking at on the stage. The stage is easy to remember because it's where the show is happening. We have the course adjustment, which will move our stage up and down. Before you put a specimen, a slide on there, you want to make sure that the stage is all the way down. So you can see it's going to be raised up as you bring it into focus. Okay, course adjustment is only used with the red ring, the smallest one that we have. Okay, let's talk about a couple other things that are here. We have our revolving nose piece. This turns from power to power. Okay, so let's talk about how you know magnification on the microscope. It is a combination of the amount of magnification in the eyepiece, which in this case is 10, and the amount of magnification in each lens. So on the very smallest lens here, which is always where you start, 10 times 4 is 40 times magnification. That's the red ring, and that's usually standard in science. Then we have our yellow ring, which is 25 times 10 is 250. Then we have the blue ring standard, that is 40 times 10 is 400. So when we look at the three objectives that we have, we end up with 40, 250, and 400 times. Now often you're going to find an advanced microscope that has a white ring. This is for oil immersion, which is different than using a slide with a slip cover. It will have no slip cover and will be using oil, and you can get up to, with this particular white ring, it's 100 times, so 1,000 times magnification. All right, so you will always start on the lowest power, rotated into the back. Some other adjustments that you may need to pay attention to is the stage clip right here. This moves a little lever that allows the glass slide to be slipped in and held properly in place. So when it's properly in place, you can see through right here that where the light is coming through is not going to show me anything. And I use the stage adjustments to move this to where the specimen is on this particular slide, which happens to be right about there. Okay, another thing that can be very useful is what's called the diaphragm. The diaphragm is usually a lever. It could be found anywhere around the stage or under the stage. It just simply moves like this. And what that does is it changes the level of light so that you can look at the cells or whatever you might be looking at in a little more detail. As you shift the light, you'll see other things. So the diaphragm is there. Sometimes it's on the side of the stage. Sometimes it's out in front. So it just depends on the microscope that you have. And remember that when you are done with the microscope, you need to make sure that you return the glass slide to your teacher. You make sure you turn it off and you return it to low power. That's very, very important. And when you unplug it, remember to hold it by the plug. And one other detail here, you want to use the special lens paper. You want to use the special lens paper to clean any lenses. In fact, you might want to simply ask your instructor to help you. The lens paper is special and it's used to clean any of the lenses that have the glass. And this way they will not be scratched. And you need to be very gentle because the lenses are fragile. And please follow your instructor's information that they may have for sanitizing your microscope. They have special wipes for you to use to make sure your microscope is clean for the next person to be putting their hands on and putting their eyes to.